Hello, good morning students. Now we'll start with the digestion of proteins. Basically, proteins in the food, it involves the milk, the pulses, the meat, the egg, and the proteins, they are broken down into the amino acids. The digestion of proteins takes place in the stomach in acidic environment with the help of HCl, in the duodenum with the secretion of pancreas, and the small intestine. The proteins, they are taken into the body, as I've told you, from number of dietary sources like milk, pulses, meat and egg. The pepsin secreted by the stomach, it catalyzes the cleavage of covalent bond in protein to produce smaller polypeptide chains. The gastric pepsin digests as much as 10 to 20% of the total ingested protein. The mucosa of the stomach, it has gastric glands. The gastric glands have three major types of cells, namely mucus cells, which secretes mucus, peptic or chief cells, which secretes proenzyme pepsinogen, parietal or oxantic cells, which secrete HCl, and intrinsic factor, which is essential for the absorption of vitamin B12. The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with the acidic gastric juice of the stomach by the churning movement of its muscular wall and is called chyme. So chyme is acidic food in the stomach. The proenzyme pepsinogen, on exposure to HCl, gets converted into pepsin. Pepsin then converts protein into proteoses and peptones. The mucus and the bicarbonate present in the gastric juice plays an important role in lubrication and protection of mucosal epithelium from the acidic HCl. HCl provide acidic media pH 1.8 optimum for the pepsins. Rennin is a proteolytic enzyme found in the gastric juice of infants, which helps in the digestion of milk proteins. Prorennin is converted into rennin plus inactive dipeptide with the help of HCl. Casein, with the help of rennin, change into paracasein, which is soluble, and calcium salts forms calcium paracaseinate. Pepsin acts on calcium paracaseinate and converts it into peptones and proteoses. The bile, pancreatic juice, and intestinal juice are the secretion released into the small intestine. The pancreatic juice and bile are released through a hepatopancreatic duct. The pancreatic juice contains inactive enzymes like trypsinogen, chymotrypsinogen, and procarboxypeptidases. Pancreatic proteases, all secreted in inactive forms, digest peptides into amino acids. Trypsinogen is activated by enterokinase, secreted by duodenum into trypsin, which in turn activates other three enzymes, that is chymotrypsinogen into chymotrypsin, procarboxypeptidase into carboxypeptidase, proaminopeptidase into aminopeptidase. So, digestion of proteins begins in the stomach in the presence of HCl and pepsin. The HCl, it plays a very important role. It denatures the protein, it decreases the pH, it activates the pepsinogen, kills the bacteria, and helps in the absorption of vitamin B12. Pepsinogen is converted into pepsin and converts proteins into peptones and peptides. Pepsin shows autocatalysis, that is, once it is formed, it can further convert pepsinogen into pepsin. So we can summarize that in the stomach we have the enzyme pepsin, which converts protein into small peptides. In the pancreas we have trypsin, which act on peptide bond, and basic amino acids like arginine, lysines and histines, they are separated, their bonds, they are broken. It activates trypsinogen to trypsin, pro-carboxypeptidase to carboxypeptidase, pro-elastase to elastase, pro-aminopeptidase to aminopeptidase. Chymotrypsin, it specially acts on peptide bond, contributed by aromatic amino acids like phenylalanine, tyrosine, tryptophan. Carboxypeptidase and elastase, they act on carboxy terminal amino acids. In the small intestine, we have got two important enzymes. One is aminopeptidase, which cuts the amino terminal amino acids, and dipeptidase, which act on dipeptidase and release the free amino acids. So when we look at the digestion of proteins, it takes place in stomach, in small intestine, duodenum of small intestine with the help of pancreatic juice, and with the help of intestinal juice in the small intestine. So the end product of the protein digestion is amino acids. Now how the absorption takes place in the case of proteins. Now we have got amino acids and di and tripeptides in the lumen of small intestine. The amino acids, they will bind with the transporters in the presence of 2 sodium and enters into the enterocyte. From there, by facilitated diffusion, they enter into the blood vessels. Similarly, di and tripeptides, with the help of transporter pepti1, enters along with hydrogen ion into the enterocyte, where it is converted by proteolysis into amino acids, and then these amino acids, they are through facilitated diffusion enter into the blood vessels. So thanks to www.google.com for providing us various pics so that we can explain our topic well. Thank you.